Well, thanks, Peter, and thank you everyone for joining in today. So I'm Bill Ketelbey, CEO of Actinogen Medical. Actinogen is a small biotech undertaking the development of a series of compounds developed at Edinburgh University. These compounds are very specifically designed to get into the brain to inhibit a particular enzyme, the 11-beta-HSD1 enzyme, and moderate the production of cortisol. Cortisol is the stress hormone, and what's been recognized in recent times is that persistently raised cortisol over time is deleterious to the brain, causing cognitive impairment and even structural brain damage. So our drug is designed to inhibit cortisol production in the brain and enhance the cognitive impairment and hopefully slow down the actual structural damage that's occurring to the brain. The mechanism of action of our drug is depicted in this image here. In the neurons in the brain is the enzyme, and our drug inhibits that particular enzyme. We've done a series of studies in recent years that have really demonstrated the true proof of concept of the development of our drug. And that's demonstrated in this slide here, where about a year ago, we showed that at 20 milligrams a day, our drug improved cognition in a healthy elderly population. We've got very good target occupancy data that very clearly demonstrates our drug adequately gets into the brain, binds to the enzyme in the brain, and then from another series of studies, we've demonstrated very good cortisol inhibition. So what we've got is proof of the design principles that underpin the development of our drug. A drug that gets into the brain, binds to the 11-beta-HSD1 enzyme, inhibits cortisol production, and improves cognition. So we're confident now that we've got the evidence necessary to move forward into the next series of studies, and that's going to be demonstrating the proof of concept of our drug in a patient population. Now, just to show you some of the data from our earlier studies, this is the XanaFX data where we demonstrated cognitive improvement with our drug in a healthy elderly population. Here we can show very clear separation of the curves. The details not necessary, but just to demonstrate that there's statistically significant enhancement of cognition, an improvement that occurs by week 2, week 4 of therapy. So a very rapid response, and a response that's persistent out to the end of therapy. And this data replicates exactly what we'd seen in our earlier animal models and our earlier human models. So we are very confident that what we've got is a demonstration of the efficacy of our drug at 20 milligrams a day. And as I say, we're now confident with moving forward into a patient population. And so we're initiating a series of phase 2 studies early next year. The flagship indications, or the flagship indication, is Alzheimer's disease. Another very important indication that we're undertaking a trial is Fragile X. And then we have a series of additional studies where there is a lot of interest in researching our drug, just demonstrating the breadth of potential application of our drug through the inhibition of cortisol in the brain and the enhancement of cognition. But Alzheimer's disease is really the flagship indication, with Fragile X a secondary indication, but a very interesting opportunity as well. Now, the reason we've chosen Alzheimer's disease and the reason we've chosen mild cognitive impairment in Alzheimer's disease is because of the enormous opportunity it presents and the huge unmet medical need there is. Mild cognitive impairment is the very earliest stage in Alzheimer's disease. As you progress down the disease into mild, moderate, and severe, you're talking about a more and more compromised brain, and a brain that is less and less responsive to available therapies. In fact, the only therapies, few that there are, that provide any benefit are in mild cognitive impairment. And so the whole trend is towards diagnosing earlier and treating earlier in the hope that you have a more responsive brain that is more responsive to therapy. Now, we, from our XanaHES trial at 20 milligrams a day, have already shown a very good, solid, robust cognitive signal, as I showed to you earlier. And the important thing is this population, this healthy elderly population, sits right alongside the mild cognitive impairment population. And it's very likely that some of the patients in the XanaHES trial could reasonably even have had undiagnosed mild cognitive impairment. So when our advisory board saw the data from the XanaHES trial, they were very excited, and they saw the huge opportunity that presented to undertake a study with Xanamem in mild cognitive impairment. And that is the study that we are initiating next year. A huge opportunity for us. So this is the trial design. Now, we've undertaken a number of studies in past years, and medical research in Alzheimer's disease has evolved substantially in recent years, and all of that knowledge, all of that learnings, all of those learnings have been built into the trial design for the XanaMIA trial going forward. So we're going to enroll an enriched population, a population enriched with biomarkers to ensure that we're not just using cognitive entry endpoints, but biomarker entry points as well. We're going to test a population 65 and above. Now, 8% of 65-year-olds have mild cognitive impairment, 50% have raised cortisol, so a really increasingly at-risk population for those above 65. We're going to study 72 patients, which is 50% more patients than our very successful XanaHES trial. And we're going to dose for 24 weeks, which is twice as long as we dosed in the XanaHES and Xanadu trials. So providing our drug with a real opportunity to demonstrate its efficacy. Most significantly, we're going to dose at 20 milligrams a day, but in a divided dose. So 10 milligrams twice a day, which from our target occupancy data, we've been able to demonstrate is the most effective dosing regimen for our drug to hopefully demonstrate the true cognitive benefit of our drug in Alzheimer's disease. Understandably, we're going to have cognitive endpoints in our study, but importantly, we're going to include the cog state endpoints that I showed you earlier from the XanaHES trial that very elegantly demonstrated the cognitive benefit of our drug at 20 milligrams a day. We are going to include, obviously, other cognitive endpoints, but very significantly for this study is we're going to include numerous objective biomarker endpoints. These are endpoints, biological endpoints, blood measures, cerebrospinal fluid measures, imaging measures, interstitial fluid measures, all of them providing biological measurements of not only the disease, but the progress and the impact of the disease over the period of the trial, hopefully demonstrating the true treatment effect of our drug, not only on the cognitive endpoints, but importantly on these biological endpoints. And that's what is going to strengthen our study so much more substantially. Big pharma, potential big pharma partners and regulators are looking for not just cognitive endpoints, but biomarker endpoints. And we have built these into our study very substantially going forward. So very key learnings have been built into this trial. And we look forward to initiating this in the first half of next year, completing it within 24 months. And I'm very pleased to say, following a successful capital raising over the last month or so, it is now fully funded. The second trial I want to speak to is Fragile X syndrome. This is due to a mutation on the X chromosome. and presents in early life with developmental problems. It is the most common cause of intellectual disability, but patients present with a range of symptomatology, behavioral problems, cognitive problems, speech and language problems, but significant anxiety. Anxiety is one of the most significant problems presented in these children at the age of 3 to 5 when they're diagnosed. What's been shown is that a large number of these behavioral anxiety and cognitive problems are associated with raised cortisol. And having demonstrated the efficacy of our drug against cortisol in the brain, we're hopeful that we will be able to show a benefit from Xanamem to treating these symptoms associated with Fragile X syndrome. Most significantly, there are currently no therapies to treat this disease, so huge unmet medical need that we're addressing. Equally, this is a Fragile X is recognized as an orphan drug, and potentially our drug could be designated as an orphan drug to treat an orphan disease, and potentially even a rare pediatric disease designation, attracting very substantial benefits in time and development and cost. And importantly, the data that we demonstrate in Fragile X potentially could open up a huge array of future study with our drug in pediatric populations, not least of which is the potential for autism spectrum. Now, the design of the Fragile X trial, it's a proof of concept study. The first time we're dosing children with our drug. So obviously we're starting in the older age group of children to define the safety and importantly efficacy in this population. It will likely be a smaller population than this, so perhaps only about 30 patients. Importantly, is being run by the Murdoch Children's Research Institute, an institute, one of the world leading institutes in pediatric research, and an institute that has been undertaking a huge amount of Fragile X research over the years, and institute that is particularly interested in running a study with our drug to demonstrate the potential for the efficacy of Xanamem in the Fragile X population. Again, this study is expected to initiate in the first half of next year, and because of the size of it, complete within 12 months. So a very rapid study to turn around, and equally pleasingly is fully funded. So both these studies, both the Alzheimer's trial and the Fragile X trial, initiating in the first half of next year, we hope. The Fragile X trial completing within 12 months, and the Alzheimer's trial completing within 24 months. So an exciting year ahead of us, an exciting opportunity for the company, where we're initiating on the back of the very substantial data that we generated in the XanaHES trial and our target occupancy trial. On the back of that data, initiating two very exciting trials in mild cognitive impairment in Alzheimer's disease and Fragile X syndrome, which are both designed to demonstrate the efficacy and hopefully replicate the efficacy of our drug in these patient populations. If we can demonstrate something similar to what we demonstrated in our XanaHES trial, we have a very substantial drug and a very meaningful and important drug for these patient populations going forward. And we look forward to updating the market and the investors over the months ahead as we begin to initiate these trials and lead in fairly short order to reading out the results from these trials. Thank you very much indeed.